Good afternoon and welcome everyone. My name is Nicole Boggins. I'm a research assistant professor of biomedical science in the Charles E. Schmidt College of Medicine, and I'm also the director of community engagement and programming for the FAU Brain Institute. Today, I'm very excited to continue our Brainy Days community lecture series with Dr. Alan Levy. Thank you very much, Dr. Levy, for joining us today for your discussion on new insights into mechanisms and treatments for Alzheimer's disease. This is the third community event in our Brainy Days series, which is a month-long celebration of neuroscience that's held annually in March, where we hold events for both scientists and our community. Just a heads up to everyone here that the next segment in our community Brainy Days program is coming up this Thursday. It is Brains on Trial, what brain science tells us about the origins and limits of responsibility. At this session, prominent brain researchers and legal advocates for individuals with mental health challenges will share their perspectives on how to implement a fairer and much more compassionate legal system. This event will again be online and live Thursday from 9 a.m. till 12:30 p.m. Just a quick note for our attendees, there will be a Q&A session after Dr. Levy finishes his presentation. Please type your questions in the Q&A section. There's a button on the bottom of your Zoom control bar on the right. The chat button has been disabled, so please do not use that chat button. Instead, use the Q&A button and your questions will be answered during the Q&A session at the end. I am now going to turn this over to Dr. Randy Blakely. Dr. Blakely is a full professor of biomedical science in the FAU College of Medicine, and he is the executive director of the FAU Brain Institute Pillar. Welcome and thank you, Dr. Blakely.
Thanks, Nicole, and thanks for organizing the series and hosting us today. What I think will be a very, very informative lecture for community members. And I'll say a little bit more about our speaker in just a second. I'd like to just spend 2 seconds saying that as director of the FAU Brain Institute, I get the honor and fun of helping more than 70 faculty level neuroscientists at FAU and working with our affiliates at Max Planck and Scripps, which also have amazing neuroscience. And together, we hope to understand better brain development, maturation, plasticity, and help with aging and age-related disorders, which brings us to the topic today. Now, the Brainy Days program has been sponsored for the last 4 years by Palm Health Foundation. Palm Health Foundation is one of the major impact factors for our community in terms of brain health. And I'm pleased to be joined today by the president and CEO of Palm Health Foundation, Pat McNamara, to say a few words about the foundation.
Thank you so much, Randy. It is a real pleasure to be with you folks today. And Palm Health Foundation is delighted to continue to sponsor Brainy Days, the series. Palm Health Foundation is your community foundation for health in Palm Beach County. We exist to improve the health of all Palm Beach County residents. 4 years ago, we launched our Brain Health Initiative, where we have continued to try to encourage residents to take the steps that they can take in improving their own brain health. And under the mantra of taking care of your brain is just as important as taking care of the rest of your body. And most folks don't realize that there are many practical things that you can do to improve your brain health. So in particular, this topic today on Alzheimer's, we are really excited to learn more from Dr. Levy, and thank you for attending.
Thank you, Pat. I'd also like to mention several of our other sponsors who've contributed to our program, and these include the South Florida Science Center and Aquarium, Osher Lifelong Learning Institute in Jupiter, and the Dana Foundation. And with that, I'd like to ask our speaker to come online and I'll say a few words of introduction for Dr. Levy. This is a real special treat for me. I've known Alan now going back more than 30 years. He and I crossed paths when he was doing his residency at Johns Hopkins, and I was just getting off to a postdoc to pursue my career. And when Alan came to Johns Hopkins, he had completed an MD PhD degree at the University of Chicago with honors. And his focus in that degree really was in immunology, and it became this convergence of immunology and neuroscience, which really launched his early career, where he developed new antibodies that could detect key proteins involved in signaling mechanisms that are affected by Alzheimer's disease. We ended up having the good fortune to spend a few more years together when he and I were both recruited to Emory University in the early 90s. And Alan went on from there to stardom, really. He became the professor and chair of the Department of Neurology at Emory, a position he still holds with a couple of different endowed roles there. He's also the director of the Emory Alzheimer's Disease Research Center. It's really one of the foremost Alzheimer's research centers in the country, if not the world. He's been a prolific scientist, publishing more than 400 papers, which is really quite astounding to have that kind of impact on the field, and uncovering new molecules and pathways that contribute to this disorder. And it's just one element of recognition that he's received. He was in 2017 elected to the National Academy of Medicine. So you're really, this would be a real treat. This is a real honor for us to have Alan here today. And again, it's great to see an old friend. So thank you, Alan. I think I will turn the floor over to you now.
Randy, thank you so much for that very generous introduction. Just please let me know if you can see my screen. Just give me a thumbs up.
Yep, we can see
it. And it's really a pleasure to be here. Randy articulated our history goes way back. And I think there's one thing that he knows, and that's we've shared a passion for science and for trying to do science that makes an impact on people. And Randy has been a model throughout my career to do that. And so being able to combine science and medicine is a real joy. And it's a real pleasure for me to share some of my passion with all of you today. Because everything we do is really ultimately focused on trying to make a difference for people. And Alzheimer's disease has been my passion, going all the way back to my days as a student, as Randy just mentioned. And so it's been tremendously exciting for me to see the progress continue at an increasing pace. And it gives me great optimism. I hope to impart today some of my optimism as we're racing towards 2025. And I'll tell you at the beginning, the reason we're racing towards 2025 is because we have a national plan that says we need a treatment by 2025, and we'll come back to that in a minute. Let me just first put up here some disclosures. I'll let you look at that. I'm not going to go through them. I might mention a few things today just so you're aware of. I will mention certainly something about Biogen, and I will mention something about Cognito Therapeutics, and just so you know, have had some support from both of those. So you can take my words for what they're worth when we get to those points. So what I'm going to try to discuss with you today is first make sure everybody's on the same playing field and give you a little bit of an introduction to Alzheimer's disease as I see it, where we are with our current treatment strategies. And I'm going to then spend the rest of the talk focusing on what we are going to do to detect Alzheimer's disease earlier in the future and intervene earlier in the future in hopefully a meaningful way. And some of the initiatives I've been privileged to be involved with from the federal level include the Accelerating Medicines Partnership, which has really been instrumental in much of the work that we've been doing. And tell you how we hope to contribute to what is really a global effort, community of scientists and physicians and community to work together in a collaborative manner to bring together effective treatments. So it's pretty striking that when I go back to my days just learning science as a PhD student, people were very unclear. This is in the really 1980s, believe it or not, where people were unclear that Alzheimer's disease was even a disease. Now you see a picture of Alzheimer up there in the upper right. And he first described a patient of his who was a young woman in her early 50s who developed paranoia and memory loss. And back in those days, he not only cared for her on a daily basis, but he hospitalized her. And then when she died, he was able to apply some new techniques to look at her brain under the microscope and see for the first time these structures of amyloid plaques shown here in the middle and these neurofibrillary tangles. And so he described these around the turn of the 1900s. We've known about these pathologies in this rare case for then well over 100 years. And what we now today call Alzheimer's disease is the disease that even superficially you can see there's shrinkage of the brain, the cerebral cortex that gives us our ability to think and remember and to talk and communicate, to love. All those parts of the brain are shrinking and much smaller than they are in a normal, healthy brain. And so Alzheimer described these microscopic features of the disease, but it was really 80, 90 years later that we only began to understand that senile forms of dementia, what had often been called senility or hardening of the arteries, was in fact the same condition that Alzheimer described in a young woman. And so this really led to a revolution in insight that this common form of dementia is actually due to a disease that we now call Alzheimer's disease. So what is that clinically? We've certainly learned a lot over the last several decades since the biomedical community has turned their attention to this disorder to understand first the natural history of the disease. And what you see here is that over the course of aging and somebody who begins to develop mild cognitive impairment, as we now call it, very early signs of memory loss or very mild changes of thinking that are very hard to distinguish from those changes that normally occur during aging. And as those changes begin to get a little more serious for some people, it evolves into a mild dementia, which is when the memory loss begins to affect somebody's ability to function independently. And as that memory declines, other aspects of cognition, language abilities, insight, judgment, reasoning, visuospatial abilities, all these other uniquely, truly remarkable capabilities of our human brain become affected. And all of us fear this end of life where people end up often in a nursing home, bedbound. They can look in a mirror and not recognize themselves and not recognize family and loved ones. So it's this very end of life, which has really been burned, unfortunately, into the minds of most people when they think about Alzheimer's disease. And I hope at the end of today, we go from this sort of tragic condition to one that I hope you will share some of my optimism with, that this is really a chronic disease that starts much earlier and is something that we can and will deal with much more effectively in the future. One of the things that I've been most struck with learning over the last couple decades, however, is that when we begin to see people coming to us in the clinic with complaints of mild or moderate dementia, which is typically when people first become brought to medical attention, already at that time, as shown in this MRI scan, there's considerable shrinkage of the brain. And these big black spaces that my cursor are pointing to show shrinkage in the part of the medial temporal lobe, which involves the circuits which form memories, sort of like the hard drive for the brain for forming and storing memories. And this part of the brain becomes uniquely susceptible and begins to degenerate early in the disease process. But what has been really striking is that we now know there's about a 20-year period before anybody comes to anybody's attention of having any memory loss, during which the same pathologies that Alzheimer described under the microscope in an advanced patient are occurring in an aging individual well before they have any signs of memory loss, including both the amyloid plaques and the neurofibrillary tangles. And we can now visualize these as shown in this PET scan. We can see these things that Alzheimer and most people thought we could only see under a microscope for years. We now are developing the tools to see them in living people. So we know from these types of approaches, as we'll talk about, that this is a chronic disease, but it's gone on at least 20 and probably 25 years or more often before people come to medical attention. And so rather than thinking about this end stage, the whole field has shifted the focus towards early and early stages of the disease, where our chances for arresting the disease are going to be much greater. So one of the reasons this has become such an important global health issue is because is what's led to it being called the silver tsunami. And this is the two waves of challenge that face us. One is the wave of aging, which is shown here. And the prevalence of Alzheimer's disease increases dramatically with age. So by far the greatest risk factor for developing Alzheimer's disease and a related dementia is age alone. As you can see in this graph, once people are at the age of 85 or so, chances are 1 in 3 or 1 in 2 that they will have Alzheimer's disease already at that age. And of course, that alone is compounded by a second bigger wave, which is the growing number of elderly in the United States and also around the globe. And so here we're looking at the US population shown in these bars over time, going up exponentially as the number of people is plotted that are reaching 85 years of age. And so you can see we're down here on 2020, and you can see, we have 7 to 8 million people in that age group, and this is expected to double or triple in the years ahead. Moreover, this is being compounded by the fact that mortality rates are increasing for Alzheimer's disease. These are data that were developed by the Centers for Disease Control. And what we've seen over the last several decades is because of the breakthroughs in medical sciences and applications for public health, we're seeing dramatically reduced rates of stroke, heart disease, cancer, and diabetes, leaving people to live longer and then develop Alzheimer's disease. So this remains now the #1 feared condition of all of us. The thought that we will lose our dignity, lose our independence, become a burden to our families as a result of our older age. And so this has really prompted national and international attention because of its huge public health importance. We currently have well over 5 million people in the United States affected with Alzheimer's disease, 50 million around the world. It's the 6th leading cause of death. And the costs are already $300 billion a year in this country, and they're expected to go up to over $1 trillion in the next few decades. So again, we just can't afford this as a society, and certainly on an individual or family basis, it's even more devastating. I remember well the days in my early days as a neurologist taking care of patients with Alzheimer's disease, how excited we were when we had the very first FDA-approved treatment for Alzheimer's disease. Little did I know that now we're nearly 20 years ago since the last drug has been approved, and the drugs that we were so excited about several decades ago when they were first introduced, in retrospect, are just nowhere near as adequate as we need to. What they evolved as treatments that made sense at the time, but we've learned so much more about the underlying cause of the disease that we're going to talk about today, that the new direction now is to find the first disease-modifying treatments that don't target the symptoms of memory loss and thinking changes, but rather the underlying disease itself, so we can slow it and then ultimately prevent it. And to address this, Obama signed into law in 2011 the National Act for Alzheimer's Disease called NAPA, which is shown here. And that's where the title for this talk came, that we're racing to 2025, because the first goal of that, as we'll talk about, was to develop an effective treatment for Alzheimer's disease. And as a result of that being signed into law, there was a major priority to increase the research emphasis. And so you've seen here just over the last few years, the research funding was around $500 million a year from the NIH. It's now over $3 billion a year, which is dedicated to Alzheimer's disease. Still, we're far underfunded compared to where we need to be, but there's certainly been tremendous progress as a result of that investment. So in terms of where we are currently with treatment, we do have several FDA-approved drugs. As I mentioned, they've been around with us for over 15 years now. They act on some of the chemicals that are important in memory. Some of the names of the drugs, the generic and the trade names are shown here. We don't need to go into those, but they're standard of care. They help many people some. They cause some side effects in some. Um, and it's important to realize that the drug treatments are only a small part of the whole treatment plan. We're going to talk about many of the lifestyle issues that influence the trajectory of Alzheimer's disease. I think it continues to be far underappreciated that this is a family disease. A person who's affected by Alzheimer's disease often will have the greatest impact on their loved ones and the caregivers taking care of them. And those caregivers need to be able to get the support they need. And there's a lot of reason to convey and confer hope and encouragement to everybody to keep them engaged, to help them do well. And again, the greatest challenge we're going to talk about today is why we have no disease-modifying treatments. So what we've seen is that over the last 20 years, as the research has continued to accelerate for Alzheimer's disease, one of the first major theories of Alzheimer's disease evolved around these amyloid plaques that Alzheimer discovered, over 100 years ago. And the theory has been that this amyloid plaque is an accumulation of a peptide or a protein substance, this beta amyloid peptide, and that as it accumulates, it becomes injurious to the brain, that it's a toxic substance which causes this neurodegeneration. And there's lots of reasons why this has been a popular theory, and many good reasons. And what's happened is that became the major focus for drug development, with the idea that if this plaque could be prevented from building up or removed, it would lead to a treatment for Alzheimer's disease. I don't expect you to read this table here on the right, but what it shows you is a recent review of all the clinical trials that have been done over the last 20 years that have targeted this amyloid plaque. And from a variety of different strategies, from a series of different treatments that block the production of it, to immunotherapies that help clear it out, and others. And the only thing to see in this whole table is that every single one of these clinical trials failed.
Hey, Alan,
Yes?
I'm not seeing the table.
Oh, you're not?
It didn't advance, I think.
Let me go back.
Let's see if we can just make your slide move.
I just went back and forth. Is that...
No, it's still stuck on the one that says Alzheimer's disease and ADRD management.
Let me stop
just
sharing.
restart. Yeah, that's a good idea.
Okay, what do you see now, Randy?
Let's see. What I see is it doesn't show your screen. It just has a black screen, so we don't see your actual screen at this point.
Do you want me to rejoin, or I can stop sharing? Let me try to stop sharing again one more time.
Why don't you try to, stop sharing and share one more time. Let's see where that takes us. Okay, you're back.
Back?
But you're on a different slide. You're on a different.
There we go.
There you go. You got it now. Thank you.
Yeah, please feel free to interrupt if any more problems. So what I was saying is that we've had a litany of failed clinical trials, and they're all listed here in this table on the right. And so what has been striking is that a variety of different approaches to remove the amyloid have failed to lead to any improvement in the dementia. And what's even worse is that a handful of these treatments actually made people's memory and thinking worse. Not only did they not improve the situation, they actually made it worse. So it's really made all of us who are involved in the field scratch our heads with great concern that we need to think a little bit differently about this. It also led to several major drug companies realizing that they couldn't afford to take the risk to stay in this. This series of failed clinical trials that I just mentioned has really been devastating for drug companies being involved in this. And it's led to a lot of attention. It's grown. We've seen Pfizer, for example, drop out of their specific efforts to develop drugs for Alzheimer's and Parkinson's. We've seen Janssen lose some enthusiasm. And let me just spend a couple minutes on what is probably something that many of you know about, which is a really remarkable story about Biogen. And they've had one of the treatments one of these immunotherapy treatments that has an antibody that attacks the amyloid plaques in the brain. And what happened is there was a reversal. There was an early phase clinical trial, which was done. This was what's called a phase 2 study. It was a small study, which was published about 5 years ago now. And what you're looking at is its early experience with this immunotherapy called aducanumab. And what you're looking at is these PET scans, which show in bright red and yellow amyloid, the amyloid plaques that are developing in the brains of these individuals. And this column on the left shows 4 different individuals at the beginning of the study, all of them with large amounts of yellow and red showing the amyloid plaques accumulating in their brains. On the right are 4 of these, the same people shown after 1 year of treatment with this antibody treatment. See, this person on the top received a placebo, whereas these other 3 fortunate individuals received increasing doses of the treatment. And what you can see, which we all saw for the first time when this paper came out, was really tremendous excitement that it was possible to remove the amyloid from the brains of individuals once they develop it. That was a huge step forward, and we were all very, very excited in the field. And what was even more exciting is the higher the dose, the more the amyloid was removed, which is shown in this figure on the upper right. And then most critically is the question of, did the drug have any impact on the clinical progression of the disease? And here the answer again seemed to be in this early phase trial, yes, there seemed to be some slowing of the rate of the decline of dementia. So again, this is an early phase study, and what happens then to get a drug past the FDA, one moves from a phase 2 small study to design a better trial of many, many more people. And that's what Biogen did, and that's where a lot of the interest and controversy has arisen over the last few months. And so they did 2 large trials of testing the drug in 3,000 individuals. One study was called Emerge, and one study was called Engage. And I'm just going to summarize. I'm not going to go through the details, but the high level is both of these clinical trials showed that the brain amyloid was reduced substantially from the brain, just as it was in that earlier phase study. And they also now were able to visualize the neurofibrillary tangles. This is a PET scan with a tau tracer, as it's known. And here you can see in the temporal lobes, these yellow and red regions down here, where the neurofibrillary tangles developed. And in this trial, in these trials, there also was a good evidence that reducing the amyloid also had an impact on reducing the amount of neurofibrillary tangles in the brain of people. That's pretty terrific. You know, that's something that we've all spent our careers hoping we would be able to see. And then the one positive trial called Emerge, it looked like there was actually less decline in terms of cognition, so that the progression of the memory loss was less severe in those that were treated with the drug. And most importantly, their functional abilities were also better. In fact, those receiving the higher dose of the immunotherapy had 40% improvement compared to those untreated in terms of the longitudinal change in their functional decline. The challenge came when the sister study of this called Engage, which was extremely closely matched in terms of the design of the study, for reasons that still are pretty much controversial and unclear, gave some different results. So here, there was no difference that was significant in the rate of change in memory loss over those treated. And there was a little bit difference in functional decline, but it wasn't significant. So we have a case where one study was positive and one was negative. So the FD, what happened is Biogen actually stopped this study early and said, because of these results, it looked like they weren't going to find a positive signal, and they stopped the trials. And then after further analysis of these data, they went back to the FDA and have requested consideration as to whether this drug will be approved or not. We're all on the edge of our seats because in June, the FDA will be making their decision. And that, again, I'll be happy to talk about this more in Q&A, but watch the newspapers in June for this decision because it very well could change our world in a variety of ways if this becomes the first disease-modifying treatment for Alzheimer's disease. So at least now we don't have any. We remain hopeful, but the question has become to the field is why haven't we been more successful? And there are a lot of reasons that I don't need to get into. The most important one that has inspired much of our interest has been the question of what else is happening in the brain. There's much more complexity in terms of the change that's happening in the brain, in addition to those two things that Alzheimer's has seen under the microscope 100 years ago. We have completely new tools and insights to understand in a very unbiased way the changes that are occurring, not just whether they occur under the microscope or not. And so we've been fortunate to participate in some of the research that is trying to discover some of these other mechanisms that might be involved in Alzheimer's disease. And again, this is a summary that I put together on this slide to illustrate a large body of work from thousands of researchers around the world over the last several decades. And what we have collectively learned is that this disease is much more than amyloid and tangles. First of all, this is a largely genetic disorder. We now understand the disease is about 70% inheritable, so to speak. So genetics plays a very large role in this. But we also know that environment plays a very important role together with genetics, as does lifestyle. And how did those factors converge to change things in the brain? They certainly do. We are learning influence the amount of amyloid that gets produced. We're learning that beyond amyloid, there are changes in the blood vessels and the blood flow in the brain. We're learning, as we're going to talk about a bit more, that inflammation is probably another underappreciated process which is occurring in the brain, which you don't see under a microscope, but nonetheless is probably playing a very important role in why the disease is getting worse over time. We know that there are changes in the brain's metabolism, which are also influencing the process, in addition to the tangles. And then ultimately, all these processes are probably converging in some way to cause the brain cells themselves to be injured, and for the brain cells and their connections to be lost, to cause the memory loss and dementia, which this disease is all about. And again, just to reiterate, much of this is happening during these 20 years or so prior to the first signs of neuron and synaptic loss when memory loss begins to appear. So again, the national plan was highly motivated by that perspective, which I just shared with you. And I'm going to focus mostly on this first goal, which we want to achieve by 2025, and this NIH mechanism called the Accelerating Medicines Partnership for Alzheimer's Disease, which has taken one of many strategies to try to open up our understanding of the disease mechanisms and use that information to inform new treatment approaches. And again, when we drill down, I know many of you are not scientists, and I'm not going to expect to get into details here. But for us to go from understanding that genes and environment are contributing to Alzheimer's disease, we need to get a much more precise understanding of what's happening. We have 20,000 or so genes that we each inherit, one copy from each of our parents. And is trying to sort out among these 20,000 genes, which ones are playing the most likely causal roles in contributing to the genetic risk of Alzheimer's disease. Similarly, we have an environment and lifestyle which are playing a role. We need to understand from large epidemiologic studies what these factors are that are contributing. And so all those types of research studies have been ongoing with great pace over the last couple of decades and continue to inform us now that the genetics, there are well over 30 genes now which are contributing to the risk of Alzheimer's disease. There's a early onset form of Alzheimer's disease where individuals develop the disease like Alzheimer's first patient in their 40s or 50s. And these tend to be a very, very strong genetic cause of the Alzheimer's disease, which is why they occur at such a young age. And this has really fueled the hypothesis of why amyloid plays such a central role. This is the actual gene called APP that makes amyloid beta peptide. But when we look at these other genes and environmental or lifestyle factors, we get clues for the first time about a variety of different processes other than amyloid, which are things of growing interest for scientists to develop new treatment strategies for. For example, some of our genes clearly point to the role of our lipid metabolism and the role of our blood vessels in the disease. And this is also borne out in some of the factors that are emerging from epidemiologic studies. For example, high blood pressure, high cholesterol, diabetes are all major causes of cardiovascular disease risk factors. They also are shared with this risk for Alzheimer's disease. Similarly, inflammation has emerged clearly from much of the genetics that there are genes linked to the immune response and inflammation, as well as clues from epidemiology that these other processes are playing an important role in the disease. So again, to get beyond sort of a global understanding that these processes exist, to get to a much deeper understanding, the NIH started this program called the Accelerating Medicines Partnership Program a little over 5 years ago. And it was a partnership formed with industry, those same industry leaders that had invested billions of dollars, all going for targets, the drug targets for amyloid or the neurofibrillary tangles. They decided that no longer should they all be doing the same things in their own closets hidden from everybody, but they should work with the NIH and academic groups at universities to understand the disease better as a first principle and to better expand the targets that might be emerging for future studies. And the strategy that this group evolved with was to start with human brains. Rather than trying to study something in a mouse or a cell culture dish, the starting point had to be human brains because there's something uniquely human about Alzheimer's disease. For some reason, even non-human primates don't get Alzheimer's disease as far as we know. Mice and other animals don't get Alzheimer's disease. So because it's such a uniquely human disease, it's important to start with understanding human biology. And the goal was to understand as much as we could about the brains from those 2,500 individuals during life and studying the brains after death through the genetics and all the ways that the genes express themselves through the RNA and protein molecules that are the building blocks of the brain tissue themselves. And then the goal was to take all of this large amount of data that is emerging from these studies and work together with special data scientists that are experts in taking large amounts of data and reducing that to bits of information that are more manageable and interpretable to help us prioritize understanding of what's happening in the brain. And so you can see here, this is a consortium of universities that have been participating in this program, and I've been fortunate to lead our team at Emory in Atlanta, where we've been spending the last 15 years developing methods to study the proteomics. So to go from the genes to the proteins is really where we've been focusing our efforts. We've been able to, really privileged to do this for the entire consortium now. And so the goal of our studies has been to take all these human brain tissues and to understand for the first time the proteins that are present in the brain and how they're organized and how they specifically change in association with the amyloid plaques and neurofibrillary tangles, and to use this information to understand what are the actual drivers of inflammation or the synaptic loss that we talked about that are involved in the disease. Step one. Step two is I'm going to show you we're very motivated to use this information to develop ways to be able to study the same processes in living people so that we're no longer studying postmortem brains, but of course, beginning to apply these to individuals 20 years before they would ever get memory loss. So we can identify early dysfunction and begin to get people tested and treated with the hope of future prevention. And so that's really been our roadmap for this process. So again, again, knowing this is not a, you guys all don't have your PhDs in proteomics of the brain, but I wanted to spend one example that is a type of information that comes from this study to try and explain it to you why we're so excited about it. So of those thousands of proteins that we measure, this is about 100 or so proteins that we have found. Each of these dots is a different protein and they all go by different names. And it's not, the details are not important other than to say that what we have learned is that these 100 proteins seem to behave in a very similar fashion across several thousand brains. So for some reason, some brains have all 100 of these proteins that seem to change in concert as if they're part of a concerted physiological or pathophysiologic process. And from understanding what these genes do, we've inferred that these genes are all related to the supporting cells of the brain called astrocytes and microglia, and they are related to the brain's immune response and inflammation. This is an example of how this group of proteins behaves. In one study, this was a study that was done out in the West Coast called the Adult Changes in Thought Study out in the Seattle area, where thousands of people had participated and had their memory assessed every year, and then many have donated their brains for research after death. And when we measured this group of proteins by our technology in healthy individuals who had normal memory and died without any Alzheimer's disease pathology, and looked at how they changed in Alzheimer's disease, we found that this group of proteins was markedly increased. The amounts of the proteins were increased as a result of the disease. There's a group of brains that we call asymptomatic AD. And these are individuals who during life had normal cognition, but when they died, their brains showed full-blown Alzheimer's disease pathology. So we reason that these individuals, had they lived longer, they may have developed dementia. But it also means that they were in this likely preclinical stage of Alzheimer's disease. So what we can do is we can see then that this group of proteins involved in inflammation is already beginning to act badly in the preclinical stages of disease. So it gives us our attention early. We can see that it reproduces across other cohorts, and we've now looked at 5 different cohorts from across the country to see that this is a highly reproducible change that occurs early. And we've been very interested to see that we can predict people's cognitive abilities by measuring these proteins in their brain. Pretty amazing. We can predict the amount of amyloid, plaques, or neurofibrillary tangles by measuring these other proteins related to inflammation in the brain. We've also been able to link in genes. We talked about genetics being a major causal factor in Alzheimer's disease. We can take all the genes in Alzheimer's disease and find that they map into that one group of inflammation-related proteins. So that's the type of work that's going on a very basic science level. And what I'd like to do is now switch gears and talk with you about the remaining time about how we're beginning to use that information to guide early detection. So we need to be able to diagnose people earlier, and then how it's informing our strategies for early intervention. An overarching goal is, of course, to detect people earlier, start treatments that can push out the disease process, and let them hopefully die of natural causes when they're 100 before they end up on the slippery slope of dementia. So there has been a lot of progress in the field trying to detect Alzheimer's disease. 10 years ago, we used to tell people in clinic that you can't diagnose Alzheimer's disease while a person's alive. It can only be done at autopsy, just like Alzheimer himself did. that is no longer true. We now have PET imaging capabilities as shown here. We routinely do spinal taps, a very safe procedure to let us measure a couple of tablespoons of clear water-like spinal fluid and measure the chemicals as a way to diagnose the disease. And we're also working on a whole new generation of other tests that I'm going to just share a little bit of the progress in the field with you and our work with you. So we already have several different agents now to let us look at the amyloid plaques by PET scans and as well as the neurofibrillary tangles. So these two hallmark pathologies are easily measured. The problem is they're extremely expensive. At Emory, for example, it may cost $15,000 and insurance companies don't reimburse it and Medicare doesn't pay for it. So it's too expensive and clearly not everybody has PET scan around the world. So it's not very available. So I don't view this as a scalable method for diagnosis, but it has been a very important research tool as we've done clinical trials. Because of the expensive PET scans, we've relied on a long-standing neurologic test where we measure the spinal fluid after a spinal tap. This has been a test that we've used for hundreds of years to diagnose meningitis, hydrocephalus, all sorts of things. And so for us, this is a routine test in our neurology clinics. And we can measure the chemicals that come from those amyloid plaques and neurofibrillary tangles and get really a precise diagnosis using the spinal fluid. That task, of course, the test is a little bit limited that many people still don't want to undergo a spinal tap. The fear of it is understandable. Again, it's something that can be done very safe, easily. Usually our patients and research participants ask if we've done it yet by the time we're done. It can be done in about 20 minutes. But nonetheless, it's something that takes expertise and time that we need to do better for a disease of the magnitude of Alzheimer's disease. For that reason, we launched something that we call the Emory Healthy Aging Study and the Emory Healthy Brain Study about 4 years ago. And this was through a generous community foundation, the Goizetta Foundation in Atlanta, decided to partner with us. And they said, what would really be transformative for the field? And what we said is we need to be able to diagnose our patients earlier in that preclinical stage and do it quickly, non-invasively, and understand the complex biology that's happening. And so we've now recruited over 25,000 individuals who participate in an online study. We have NIH funding to expand our 1,500 up to 3,000 people that come in every other year for detailed assessments from brain scans to PET scans to cardiovascular testing. And then we do genetic studies and spinal fluid studies to try to get a data-driven approach to understanding how we can better predict who's going to get Alzheimer's disease and when. I showed you the work that we're doing with the protein level work. This is that same inflammation protein module I showed you. Now, one of the things we've been able to do is take these same protein targets that we identified in brain and measure them in spinal fluid. And so we now can measure this network that we're seeing related to inflammation in the brain by measuring in living people the spinal fluid samples. As I think I'll show you in a few slides, we think this is going to be an important way to help identify people who might have more inflammation, who might be candidates for inflammation-related treatments. It's funny, in our healthy aging and brain studies, we put people through hours and hours of testing, including the spinal taps. The thing that our participants tell us they hate most of all are the memory tests that we've had them. The traditional pencil and paper memory tests typically take 45 minutes. And they've become just the nemesis of our participants as well as us because people just don't like having their memory challenged. So we decided to take advantage of a new strategy where we could use what has been learned about cognitive processing in our non-human primate studies at Emory, where we can track monkeys' memories by looking at what they looked at. I'm not going to go through the details, but we've been able to adapt a test that has been used in monkeys so we can have people looking at pictures without even telling them that they're looking at a memory test. And then we change the pictures after a couple minutes and remove an item. So for example, here you're looking at these two kids with a suitcase. After 2 minutes, the person looking at this image will see the image again, but this time the suitcase will be removed. And so if the person remembers this, even subconsciously, they will look at this area because there's been something that their attention is, notices, and their memory system is engaged with. So we can predict memory performance pretty accurately just by having people look at pictures passively over a couple minutes. So we're now in the process of deploying this in more of a smartphone app so we can have a more widely available test. One of the exciting partnerships we developed was with a company called Neurovision out in California. There are a few different groups that are doing this. There's been the idea that the amyloid plaques which develop in the brain may actually be developing in the eye as well, in the retina. And so the retina is actually comprised of neurons. So it's a brain-like substance in our retina, which receives light and transmits that information back to our brain. So it really is a neural structure. And what you're looking at here is a picture of the retina in an individual. And these little white spots are amyloid-like spots that are lighting up after the person drank a milkshake full of a dye, which is actually a spice, which binds to the amyloid plaques. So a very non-invasive test gives us the ability to now have promise for measuring amyloid in living people just through an eye test. The holy grail for the field has been a blood test for Alzheimer's disease, where we always longed to be is to have something like a cholesterol equivalent test that we all have measured to help monitor our risk for a heart attack or stroke. And it's also a target for measuring to know that the statins are needed, that the statins are working to lower our cholesterol for those in need. So this is something that we didn't think would be so feasible, but over the last year or two, several different groups have evolved different tests that look very promising for measuring amyloid in the blood of living people as a surrogate for measuring the amount of amyloid plaques in the brain. In fact, one of these is a company that now has a marketable test which is available for doctors to order. And what's really exciting is that now there's a blood test emerging again from several groups, which seems to be able to measure with some degree of accuracy the neurofibrillary tangles present in the brain by measuring the same protein derivative called tau in the blood. And so this test seems to be very good at distinguishing, even in the preclinical stages. This is a group of people here compared to the controls here. So people that are in the preclinical stage of Alzheimer's disease who have amyloid in their brain already are showing up a positive blood test. And it gets worse as the Alzheimer's disease evolves over time. And this just shows you how this blood test for the neurofibrillary tangles correlates with the actual PET scans showing the neurofibrillary tangle burden in the brain. So again, a very promising association between the blood being able to measure what's actually present in brain. So I'm very confident that the progress in early detection is going to be reaching all of us in clinical medicine, I think in the next year or two, and we're going to see continued progress. And so I think one of the biggest challenges is being able to identify those people early. Right now, there are 48 million people in the United States walking around with amyloid who don't know it because they don't have any symptoms. We need to detect those people. And then, of course, let's focus the last few minutes on the reason for detecting a person early is because we want to intervene early and save the brain before it gets affected by the amyloid plaques, and tangles, and inflammation, and all those other things. So it's been recognized by many that many of the risk factors for Alzheimer's disease are potentially modifiable. And so the epidemiologic studies I showed you before have identified, for example, all of these factors shown here in red as risk factors for Alzheimer's disease. Similarly, there are some factors which seem to be protective against developing Alzheimer's disease, including more education, Mediterranean or other type of diet, which is anti-inflammatory. An active lifestyle is protective as opposed to a sedentary lifestyle, which is a risk. People who have been taking anti-inflammatory drugs seem to have a lower risk. People who take cholesterol-lowering drugs seem to have a lower risk. It's early in the day for all of you, but don't forget to have your glass of red wine later tonight with dinner. That's a good protective mechanism for Alzheimer's disease risk as well. So again, the question is then, can we really modify people's risks through controlling those? And our first clear answer to that came over the last year. This has been a very important study called the Sprint Mind study. And one of the things that we all routinely should have done when we go to our annual doctor's checkup is to have our blood pressure monitored. And many of you may have had the experience where you see your blood pressure start getting up to 130, 140. You might even take blood pressure medicine and find that that's acceptable. Well, a group of researchers asked the question is, if we control blood pressure much more rigorously, if we shoot for a goal of 120 systolic as opposed to standard guidelines, which had been letting people be a little bit more flexible by keeping treatment at least under 140 or higher, they asked the question, will this be more protective? And this was from a big study of blood pressure looking at its effect on heart disease and stroke. But they followed these individuals who had participated in either standard treatment or intensive treatment for many years after that treatment trial ended. And what they found is that, in fact, rigorous attention to blood pressure control reduced the risk of normal people developing mild cognitive impairment and or dementia by about 20% over that window of 5 or so years. This has been very, very important, and I think not widely appreciated enough that we have to really pay attention to some simple things that are already doable. And these are going to be, again, things that probably should be done pretty intensively beginning in the 40s and 50s, so much earlier than I think we ever appreciated. In England, the Lancet commissions did a study looking at trying to understand how much of Alzheimer's risk actually could be modifiable by paying attention to these risk factors. And this diagram here shows the beginning of life at the top of this life curve, taking the path all the way down here towards the end of life, trying to look at those different modifiable risk factors and when they occur. And so, for example, here in the early stages of life, education alone, by improving education, can potentially mitigate a fair amount of Alzheimer's disease late in life. And I think this is probably underappreciated, would be my guess. We're learning more and more about the social determinants of health, people who don't have access to the same levels of education, even though they have as many years. The quality is important. The availability to have an enriched environment is very important. In midlife, hearing loss, interestingly, has been associated with a risk factor for Alzheimer's disease. That doesn't mean yet that we know that if you treat hearing loss, it will prevent it, but it's an open question now that needs to be studied. And we talked about the impact of hypertension already. And then late in life, there are also very important risk factors that should be modified where possible, including reducing smoking. Depression, loneliness, and social isolation are huge problems for the elderly as they lose spouses and friends. And that probably is playing in ways that we don't really fully understand yet to enhancing the progression of Alzheimer's disease with a risk. So all told, there are some estimates that maybe about 35% or so of the risk of Alzheimer's disease can be modifiable. So in the last few minutes, let's talk a little bit more about some of the other types of treatment approaches. I talked about some of the proteomics and the molecular studies that we're doing to discover new targets. One of the efforts that we are involved in now is to turn those new targets into drug-like molecules. And so we've been fortunate to have one of 2 national centers for drug discovery for Alzheimer's disease to take these novel targets and work with an international team to develop compounds for many of these new targets for which we don't have a good understanding. This is a summary from a colleague of mine, Jeff Cummings, who every year puts together an overview for all of us of all the clinical trials ongoing in Alzheimer's disease. And as of this past year, there were 121 different types of drugs being tested in clinical trials. They're all shown here. And they approach different things. Some of these are disease-modifying approaches shown here. Some of these are targeting the symptoms of memory loss or confusion and paranoia that occur as part of the disease. So what's really striking to me is it seems like a lot, but when I compare what's going on to cancer, We have 121 agents in clinical trials for Alzheimer's disease. The cancer field has over 3,000 currently in clinical trials. So this is what this tells me is that we really need to up our game. We need to be testing many, many more things as early as possible. One of the ways to accelerate this pipeline, while it takes maybe 25 years to develop a new drug, is to learn how existing drugs work and learn that they often have benefits that we might not have anticipated. Again, taking a lesson from the field of cancer research, cancer researchers have taken a variety of medications which exist and have clinical trials ongoing to demonstrate whether they're effective for cancer. These include the cholesterol-lowering drugs, anti-inflammatory drugs, antidepressants, diabetes drugs, hypertension drugs, ARBs, and beta blockers. And what's really remarkable is all of those exact same drugs, there are studies in animal models and other preclinical studies which suggest they also are attacking mechanisms which might be relevant for Alzheimer's disease. So this is, I think, again, a very underexplored approach in Alzheimer's disease, as we need to turn up the rate of discovery. In closing, I'm just going to share a couple of these new studies that we have begun in our patients at Emory, our research participants. One of the studies is one that involves a part of the brain called the locus coeruleus, deep down in the brain. Randy is very familiar with this part of the brain. It's very related to many of the things that he has been a pioneer in understanding how the molecular pathways work in these brain cells. But this is a group of cells called the locus coeruleus down in our brain cell. And we've known for a long time that they degenerate. Here is a picture of this group of brain cells in Alzheimer's disease, as opposed to all these healthy black staining cells in a control individual. And what we have learned over the last 10 years is, strikingly, this group of cells is the very first group of cells in the entire brain that starts developing Alzheimer's disease pathology. So it turns out even in individuals in their 20s and 30s, they are developing these neurofibrillary-like tangles here in the brainstem. So just think about this in their 30s, and then 20 years before that's turning into full-blown neurofibrillary tangles and amyloid, and then another 20 years before the symptoms begin. So one of the things that's exciting about the discovery of that group of brain cells is that the locus coeruleus releases a chemical called norepinephrine. It's a neurotransmitter, which diffuses and acts on other parts of the brain to carry out its biology. And it's long been studied and known that there are drugs available already, which are in clinical practice, which can influence the activity of this group of brain cells. We chose to study a drug that is out there for attention deficit disorder. And so seeing a loss of these brain cells in humans is one thing, but a variety of researchers, Michael Hennecke in Germany, David Weinschenker at Emory, has got us involved in this research and enlightened us. Turns out when these brain cells get injured, as they are in Alzheimer's disease, there's a whole cascade of changes which occur in an animal model. And so you can recapitulate some of the Alzheimer's disease changes that occur in humans in a mouse by just lesioning or causing injury to those brain cells deep in the brain. And what was really exciting is to see some other researchers, Doug Feinstein in Chicago, particularly use one of these FDA-approved drugs, atomoxetine, in combination with a precursor drug. It showed that if you give these animal models this FDA-approved medicine, you can reverse some of the problems with Alzheimer's disease. So knowing that this drug was already being used in children and adults worldwide for attention deficit disorders, we decided it would be irresponsible for us to not at least try this in humans. And so we just recently completed a study in people with mild cognitive impairment due to Alzheimer's disease, where we actually did that. We gave, you can see about 17 or 8, we started with about 20 people, either starting them on a placebo or on the drug atomoxetine. We were able to measure that the drug had its intended biologic effect on the neurochemicals in the brain and spinal fluid. And we were delighted to see that actually there were, in the spinal fluid, the measures that we used to assess the degree of neurofibrillary tangles and neurodegeneration was less as a result of treatment over 6 months with this. And we also did the PET brain imaging and showed that brain metabolism also improved in the memory circuits in the brain as a result of treatment with the drug. And we also showed that these inflammation and other related proteomic biomarkers that we have discovered in the brain, they were able to reverse or normalize many of these protein groups in the spinal fluid as well. So I view this as a very exciting direction for us to go all the way from discovery into clinical trials in humans. And then I think my very last example I'm going to show you, really excited about, because it just shows you how out of the box some of the novel approaches are going to be in the future, and why I'm so excited. Annabelle Singer is one of our colleagues in the Department of Biomedical Engineering at Emory and Georgia Tech, and she came a couple years ago after a postdoc experience at MIT in Boston. And there she had been studying the brain cells that are involved in memory, and they realized that they could influence the memory of these mice by turning on a light that activated certain brain cells or inhibited others, and that they could change the frequency that those brain cells fired to improve the memory of those animals. And what was really the dramatic breakthrough that they made was not only could they do it with electrodes in the brain of those animals, they could flash lights non-invasively, or play sounds, or stimulate the animals non-invasively to cause the brain cells to achieve that same activation. And so flashing a light 40 times a second, for example, was able to change the brain cells firing to 40 times a second. And the dramatic thing is that actually led to amyloid getting cleared out of those brains from these mice. And so here in this panel up here, these green spots are the amyloid plaques. There are far fewer as a result of treatment with this light flickering in a very simple manner. That's quantified here. So with that, again, it seemed irresponsible to not take this on. Georgia Tech is full of engineers, and several engineers came to us and Annabelle and said, you know, we can make these devices in our garage, let's do it. They actually started a company out of MIT, the founders of this discovery, called Cognito Therapeutics. So we decided to partner with them. And so we've now done a small phase 1 clinical trial using these light stimuli and sound stimuli, where you can actually have clicks going at 40 times per second, as well as the light flashing. We have our patients doing this for about an hour a day. Now we're studying to see if we can actually change the brain pathology. I have covered a lot of territory. I think I'm right on time, but I'll be pleased to take any questions in the last few minutes. This is, I hope you see a passion for me as a representative of a large field globally, and certainly my colleagues at Emory shown here are extraordinary. Much of the proteomics that I've discussed in the Accelerated Medicine Partnership has been done in concert with my colleague Nick Seafried, the Department of Biochemistry at Emory. This is a team science approach at its best. This consortium, funded by the NIH in industry, encompasses all these institutions shown down here, industry partners who have been part of the team as well. So it's been really an exciting time for us, but we know that there's a lot more work ahead. So with that, I will turn off the slides, and thank you all for your attention.
Great, Alan. What a wonderful presentation. We learned a tremendous amount. I'll turn to the questions from the audience. I'll just remind you, if you use the Q&A to ask questions, we'll go on for a little bit. We're probably going to run over the time that we had, but we'll stay on a little bit longer. One of the questions that comes up quite a bit, particularly because we see commercials all the time, and that has to do with supplements, nutraceuticals, Prevagen, you know, a number of things that are out there. What's your opinion about the science behind these kinds of highly advertised substances?
The science that has the best foundation, in my view, is the economic science. So this is a great way to make money. The industry of selling supplements is massive. I wish we had a penny for every dollar that was spent in this country on supplements to go into research towards a cure. Um, I view nature as full of incredibly powerful discoveries, right? The substances in nature are the most potent. And we know that they can be the most effective. So many medications have come from observing natural substances and learning what they are made of and turning them into drugs that can be studied to see if they're safe and effective. But nature is also full of the most horrendous toxins. And unless things are studied carefully, there's high risk that things can have detrimental effects. And so I'm all for science and all for more science when it comes to supplements. I think things we eat and take in are really an understudied area. But I really have concerns because the supplement industry is not very well regulated. So if you buy two batches of what you think is the same thing, there's no guarantee that the substance you think is in there, and there's no guarantee that it will be in there the same amount the next time you take it. So I have way too many patients that I've seen take 10, 15 supplements a day at great expense, and then find out when you stop them, they actually feel like their thinking has improved.
Right. Some questions from our listening audience here. One person asks that, as some do, that if you have a history of Alzheimer's in your family, would you recommend they have one of the PET scans if they can afford it, now that you showed? And if they, let's say they did see a large amyloid content burden, what would they do about it?
It's a great question, and I think a question that we're going to be confronting increasingly in the years ahead. Today, however, I think the answer in my mind is pretty easy. No, you shouldn't get a PET scan if you have family history. Right now, the tests that we're talking about are often research only. They haven't really been approved and demonstrated to be beneficial yet in clinical practice for an individual. Two, we're really starting with people who have memory symptoms. So if you have a family history and you have any concerns, or your family or friends have any concerns that your memory is changing more than might be expected for normal, then definitely, see your doctor. There are lots of things that can cause memory loss that are a masquerade for Alzheimer's disease I didn't have time to get into. But simple blood tests to make sure your organs are working well, your liver, your kidneys, to make sure your vitamins in your diet are getting absorbed properly. Those things are all known causes of memory loss that should be identified and corrected. So stay tuned. If you have a family history, I would say, and you're interested in research, there are studies that are increasingly being done on healthy individuals, but they need to be done in a carefully controlled environment.
Here's a question about wondering about the theory that Alzheimer's disease is a type of metabolic disorder caused by insulin resistance of the neurons and connective supportive cells, and has been called by some people type 3 diabetes. Is there any credibility to this theory?
Indeed, yeah, and that's absolutely right. The question is, the person who asked that question is 100% correct, and a lot of evidence that has linked glucose metabolism and glucose resistance, specifically in Alzheimer's disease. One of the types of PET scans that I showed you is called a deoxyglucose PET scan, which measures metabolism. It looks at the ability of brain cells to take up glucose. And it has been known for a couple of decades now that people with at high risk for Alzheimer's disease, either genetically or family history, before they get Alzheimer's disease, in those preclinical stages, there definitely is a change in glucose resistance. There are drug treatments that are being developed just for that reason. And so it's certainly a very important theory that is under study.
Here's a question about sort of the familial basis of Alzheimer's disease, and probably a personal experience here. Would you have recommendations for someone whose parent or the parent siblings all had dementia? It sounds like an increased frequency than one might expect. Should they, one, be concerned that they are at increased risk, And two, should they, use that information to go in and see someone?
Yeah, I think it's very reasonable if there's a very strong family history, particularly if it's younger individuals. So if everybody in the family seems to be getting it, but they're in their mid to late 70s and 80s, that's something that is probably not going to be won't warrant something special to be done in terms of genetics and intervention. But if people are developing the disease routinely in their 50s, for example, absolutely, I would encourage somebody to seek attention so that appropriate genetic counseling can be done with the family and testing. Those are exactly the individuals that I suspect are going to be the first to benefit from the new breakthrough treatments. There are lots of studies going on in those families with a strong genetic risk. If it's a family history where it's not young onset, is there something you should do? Absolutely. You should be paying attention to those modifiable risk factors we talked about. Particularly make sure your blood pressure is good, your diet is good, you're getting exercise that follows the American Heart Association recommendations for 150 minutes a week, and paying attention to all those other factors which likely contribute to brain health.
Given the COVID pandemic, and we're hearing about long COVID, and we're hearing about the long-term cognitive effects that some people have who've had COVID, not all, but this is something I guess is now starting to be studied. Is that something by way of an environmental influence that might be exacerbating risk for Alzheimer's disease?
What a great question. It's a question on all of our minds, right? Because we now know we're going to face another generation of individuals who has had this environmental impact. And as I hope made clear during my presentation today, Alzheimer's disease often can lag 20 years after insults or begin in the brain. And so I think we're going to have to carefully follow people. A lot of the things that we're learning about inflammation and immune mechanisms being involved in Alzheimer's disease, like we talked about, are one and the same as the immune system, the way it fights a virus like COVID. In fact, one of the theories I didn't get in today is there's a recent theory that Alzheimer's is actually associated with herpes virus, a different type of virus in the brain. So it's a great question.
I think that I see a couple of questions that sort of maybe you can help people understand. You've talked about the immune system a bit, and I think when people hear immune system, they think about antibodies, that peripheral mechanisms, but may not be as familiar with things that go on in the brain with the immune system. So maybe you could clarify a little bit what the brain has to do with the immune mechanisms.
Yeah, boy, that's a great question. It's a challenge to see how I can try to succinctly answer that, but let me take a stab at it. So, the brain is part of our body, first of all. So our immune system really has a couple different arms. Antibodies float around in our blood and help tackle viruses, for example, and bacteria when they come in. But part of our immune system are also the white blood cells, the lymphocytes that also fight cells. And those are together with cells which are scavenger cells, which gobble up the bacteria or viruses as they get attacked by our immune defenses. So part of our immune system has evolved to fight off foreign invaders. In the brain, those same mechanisms which are present to protect our periphery, that are floating around our blood, get access to the brain. So one thing that we're learning is that the brain is not as protected from the rest of the body as we once thought. And so certainly antibodies and white blood cells out in the blood can get into the brain. There are also specialized sets of immune cells in the brain. The scavenger cell is a cell called the microglial cell. And this too, they're normal part of the brain. In fact, this whole system in the brain is also involved in how the brain develops normally, just like it's involved in how all our tissues develop normally. You can't have a major organ function without our immune cells somehow being part of that process and learning what is ourself versus what's a foreign invader. And so in the brain, if there's an acute injury to the brain, like a stroke or an infection like meningitis, that immune system sees that foreign invader, that injury, and comes in and is a critical part of a repair system. So it's our first line of defense to promote healing. And that's good. And that's what happens in COVID. You know, there's this inflammation that's there to attack the virus and get rid of it. But then also, as in the long COVID or in Alzheimer's disease, if that inflammation persists, it can become more damaging. So what became first a protective mechanism turns into an injury to the brain ongoing on a chronic basis.
So I know that recently the NIH has put out a call for grants to look at the non-cognitive aspects of Alzheimer's disease, and seeing your work there on the norepinephrine system, which has been linked to mood for decades, seems to be a reasonable connection. So in the study that you're looking at, are you also assessing the mood, or do you plan to assess mood in these subjects?
We are. I've been really inspired by some of the epidemiologic studies, for example, which have suggested that depression almost has as strong a role as the strongest genetic risk factor. One of our team members, Thomas Wingo, and his wife Alicia Wingo, have been studying the genes that influence depression and Alzheimer's, and have found common sets of genes that do both. So we actually think that the genetic risk for Alzheimer's disease may actually in part be mediated by depression. And then another sort of factor like that, these non-cognitive factors, are sleep. Sleep is probably going to be one of the most important understudied areas, I would say, as something that might influence the brain in both protective and in damaging ways, depending on the type of sleep you get. So I think getting to better understand how sleep affects Alzheimer's disease and depression alike is probably going to be a clue to how we might think about treating people in the future more effectively.
Well, I think our time now has run its course. Dr. Levy has some other appointments to get to, and I hope I summarized some of the thoughts and questions for people in the audience. I would like to know what kind of wine that you recommend, but maybe we'll have a chance post-COVID to discuss that one-on-one. So it's
Absolutely.
great to see you, Alan. I enjoyed your visit, and I hope you all found this program informative. We will have the video of Alan's talk on the Brain Institute website. That's ibrain.fau.edu. And if you go to the community tab and look for the Brainy Days program, in a few days there will be a video of today's session. So I want to thank Dr. Levy again, and thank you all for joining us. And I hope to see you again, perhaps at Thursday's program on the brain on trial. Thanks a lot.
Thank you all. Thanks, Randy.
I'll see you later in the day.